Welcome everyone. So today we are gonna study neuroanatomical studies. Which brain area does what and how cells are connected. So in neuroanatomical study, we study that which area of brain does what, like what part every brain part does, plays, and how cells are connected. Two big methods are given of neuroanatomical studies. So here are two methods. First one is histology. In histology we study dead brain tissues under microscope. Second one is invasive technique. In invasive technique we test on living animal brains because of ethical issues. We listen it and stimulate it and then inject stuff and then see how the behavior changes. Then do histology to check where you hit. So and then we check the dead brain tissues under microscope to check where we inject the stuff. Okay, next is histology steps. So we have 5 steps of histology. We're gonna take a look at that. First one is perfusion. Pump saline through heart to wash blood out. Blood clots messes up slices. So basically we pump saline into heart so that the blood that is in the shape of clots will be washed out, so that taking other steps will not be confusing. Next we have fixation. So in fixation we pump formalin to freeze cells at death. We freeze the cells and the freezing level is, the cells start to die, so they do not rot. Locks protein in place. Once they cells freeze to death and they do not rot, the protein is also locked in one place. It does not move from here to there or starts to place on it. And third step is hardening plus sectioning. We freezes brain just right, then use microtome machine to cut paper thin slices. Then use microtome machine to cut brain slices, thin slices. Too hard is equal to crumbles. If the slices are too hard, then it starts to crumble. If it is too soft, then it is mushy. Fourth step we have embedding. We put brain in wax or nitrocellulose, so you can slice it thin without it falling apart, otherwise it's gonna fall apart. Fifth step we have staining. We add dyes. Different dyes light up different parts because brain is transparent without the dyes. So here we're gonna take a brief look at stains. We have 4 types of stains here. First one is Nissel blue, which is only cell bodies, only used on cell. Why? Because it shows the cell bodies is equal to soma. Soma axon. So it shows the soma, which is in blue color. And for easy memory trick, you can use Nissel is equal to nucleus, which is used on nucleus, cell body stain. Okay. Next we have wheel stain. It shows myelin on fibers, which is of brown color. Wheel is equal to white matter, myelin stain. Golgi silver, 2 to 3% neurons fully. It shows 2 to 3% neurons fully. Soma plus dendrites plus axon. Golgi is equal to ghost of whole neuron. Neuron fully, soma, axon, dendrites, all. And then we have histofluorins, glows in color, used for dopamine, noradrenaline. So it is like its color glows. It is used for neurochemicals, neurotransmitters. You can, for memory trick, FLAC is equal to highlighter is equal to flashlight for brain chemicals. Okay, next we are going to take a look at immunocytochemistry. What is immunocytochemistry? It makes antibodies for a specific protein. Okay, like any protein that is inside your body, it makes it bearable so your body can work. Okay, it makes antibodies. Recording brain activity. Now we're gonna take a look at how brain activities are recorded. I think we will take a look at in the next lecture, because it's a whole new topic. Okay,